Hi friends, welcome to Field Crop video series. In this video, I'll talk about oats. Oats is the sixth most important cereal in the world. It follows wheat, maize, rice, barley and sorghum. The systematic classification, the common name of oats is Oat Family. It belongs to family Poaceae, scientific name Avena sativa, and with chromosome number 42, that is 6x, means 6 exploidy level. It is hexaploid. Use of oats: it is used both as fodder and grain, good source of protein, fiber and minerals. Most of the oat grains is consumed as animal feed. Oats hull is a food processing byproduct, may be used as animal feed, fuel for power plants. Oat hulls are basic raw material in the production of perfural such as nylon, lubrication oils, etc. Cytology of oats: it's a natural allopolyploid which evolved through cycles of interspecific hybridization and polyploidization on combining three distinct diploid genomes. Origin of oats: it is cultivated, has evolved from the wild hexaploid species Avena sterilis. It originated in Asia Minor. Related species of oats, that is Avena sterilis, a wild red oat; Avena fatua, common wild oat; Avena byzantina, red oat; and Avena sativa orientalis, that is common side oat. Coming to floral biology of oats, the flowers are enclosed in a scales or bracts and grouped in a characteristic structure called spikelet. This entire portion is called as spikelet. Spikelets of oats are arranged in a panicle. Each spikelet has a small joint axis or rachilla which bears floret. Within the spikelet, the florets are present like this here. Floret may vary from 2 to 3. When it contains 3 floret, it may consist of opened primary floret, the outer one, then unopened secondary floret, that is this bigger one, and rudimentary tertiary floret, that is the central one. Blooming begins in the upper floret. Oat has incomplete flower because it lacks sepals and petals. Each floret consists of lemma. The outer boat-like structure is lemma and inner small boat-like structure is palea, then stigma, stamen and 2 large glumes outside. This is stamen, then bifid stigma. As we know, oat is having bifid stigma, then ovary. These are outer glumes and the connection is called as rachis. Selfing and crossing techniques in oat. The equipment required are dissecting forceps, dissecting scissors, needles, paper clips and cover bags. Selfing technique. Oat is predominantly a self-pollinated crop. In order to ensure complete selfing, the spike is enclosed in a bag. The bagging material could be cotton cloth or paper bag or cheesecloth cloth bag. Like this is a paper bag to avoid the contamination or to protect the pollen. It is the best method to protect it. Crossing techniques. First one is emasculation. In crossing, to emasculate, we have to remove the anthers with forceps. It may be made at any time of the day, preferably morning, because mornings generally better for pollination. Steps in emasculation. First, we have to select the spike for emasculation, which is still enclosed with a flag leaf sheath, means opened but not completely opened spikelet. Open the flag leaf sheath by forcing leaf edges with the help of forceps. Then cut the sheath and flag leaf at the level of first node of the rachis. This we have to cut. Then remove lateral florets and tip of the spike. Remove all the anthers with the help of forceps from all the florets. All florets on the spike must be emasculated to prevent self-fertilization. Like this it should appear, and to ensure a successful pollination we have to remove all the anthers, all the 3 anthers. Bag the emasculated spikes to prevent contamination from foreign pollen. Pollination. Emasculated flowers are ready for pollination. Pollen viability will be there from early morning to mid-morning. Stigma, that means up from 6 am to 10 am. Stigma is receptive at all the time of the day if pollen is available. It also depends on the humidity and temperature. Steps in pollination. First, to select a spike in which the anthers have begun to dehisce. Cut the top 1/3 of the spikelet to expose the anthers like this with the help of scissor. When pollen become visible, spike is dusted on emasculated one. Attach the tag with the following information: the objective of crossing, date of emasculation, date of pollination, and who has done it, either a breeder or worker. Bag and pollinated spikes. This should be covered with a pin. Problems in crossing. Pollination is more tedious to perform. Artificial seed set is low. Factors affecting seed set in artificially cross-pollinated oats are immature pollen, injury to the stigma, temperature, time of the day that pollination are made, then interval between the emasculation and pollination. Thank you. Hope you like this video. Kindly subscribe my channel Varsha Gaitonde. Thanks once again.